Yeah, thank you so much for the kind introduction and basically spelling out what Microsize has been up to over the last few years. We're building an ecosystem to accelerate drug development. So a few words about Microsize. We're a CDMO that are experts in particle size reduction. We've been in business for over 30 years, and we're one of the largest tollers in the world. We move about 600 metric tons of material through our facility in a year, and essentially our batch sizes range from a few grams up to over 30 metric tons. We work on every ingredient that goes into the tablet or the drug product. Our APIs, we can handle controlled substances. They can be highly potent. The excipients that we micronize, they could be some of the ones that are the toughest to grind, such as the celluloses. We also have a large footprint. We have over 100,000 square feet. So we have room to expand in either making labs, more micronization suites, or starting to bring in strategic partners that complement our technology. So such as the Solubility Company, where we just built a lab about 2 years ago. We onboarded them. Austin PX, we're bringing their compounder over to help with GMP capacity. And then we have the Solumatrix platform to make nanoparticles in a novel way. So our primary focus at Microsize is milling, micronization, and classification, where we have mechanical mills, such as hammer mills, as well as pin mills, to either make material into a coarse powder or a fine powder. Our bread and butter is jet milling, where we have loop mills as well as spiral jet mills. Loop mills are lower energy mills compared to a spiral jet. You have just the bottom zone of the mill that does the micronization, and the rest of the loop provides a gentle classification, as opposed to a spiral jet mill, where you have a ring of air all around, and it creates particle and particle collisions uniformly. And then typically with a loop mill, you're around 100 microns on the D90. A spiral jet mill, you can get under 10 microns. Most cases, we can get under 5 microns to support inhaled formulation products. And then we've onboarded the Solumatrix platform, where they can make nanoparticles. And this is just pure powder on powder. There's no liquid involved to serve as a media. So it's basically your API, your excipients, and then stainless steel balls to grind the material into a powder. And this powder is directly compressible into a tablet, so there's not a drying step needed. Then we have 2 types of classification. One is your standard vibratory screen, and then the other one is turbo screening, where material is pneumatically conveyed onto the screen. You have a wand pushing against that material so the screen doesn't become blind. Fine material passes through the screen, and coarse material gets rejected out to the bottom of the chute. We use turbo screening in a couple of creative different ways. We can couple a jet mill to a turbo screen. So say if we're making too much fines, we could cut the fines out. If we're too coarse, we could use that as a guard to prevent coarse material going to your product. We also use turbo screens to help control the form of your compound. So micronization tends to dry materials out. If you have a hydrate, that's something that you want to avoid. So we could use a turbo screen to separate the individual particles on metric ton scales, humidify the room, and then basically rehydrate your material after a micronization. Like I said, we have lots of space where we've been building, and so I was onboarded to build a solid state characterization lab, where I brought in image analysis with a scanning electron microscope and a polarized light microscope. We have the Microsize 3000. I've purchased a number of solid-state instruments, such as powder X-ray diffraction, DSC, TGA, DBS. And then the third theme of that lab is powder flow. So we have an FT4 powder rheometer, as well as bulk and tap and angle of repose. We implement DOE studies at the site using STADI's software. And with the addition of the solubility company, now we can offer a novel way to measure solubility and dissolution. And this will help us judge what kind of particle size we should be targeting to make sure everything is dissolved in the time that it needs to be dissolved. Multiple speakers have talked about the refined developability classification system. I was at Janssen a few years ago. That's where I basically learned pharmaceutical sciences, and this is the way I view how to narrow down your formulation space. And milling and micronization covers quite a bit of it. I recommend for even DCS1 and class 3 compounds to add some form of milling. Not that you're trying to enhance the solubility or enhance the dissolution rates, but just overall it helps with content uniformity. I have some really cool SEM images later on that shows what happens when you blend particles that are not having a size match. Bruce talked about DCS2A, where that's the bread and butter of micronization as well as nanomilling to improve your dissolution rates. And this is where micronization tends to fall off, where you need other technologies when you're entering the class 2B and the class 4 space. And this is where Microsize is bringing in other companies to help fill those gaps at our site. So we've had successful collaboration with the solubility company to help us design formulations, as well as bringing on board Austin PX's compounder. So essentially, these are the 3 other companies that are under one single roof at Microsize. And I'm going to spend a little bit of time talking about the solubility company and Austin PX. So with the solubility company, they're a small startup out of Helsinki, Finland. I went over there for 3 weeks, learned their technology, built a lab, and then brought it back to the United States. And it's been running for about almost a year and a half now. Essentially, you need a tiny quantity of API to measure solubility. We're talking about less than 100 micrograms, so it's basically preparing a slide for a microscope. If you could see too much of it, you probably have too much sample loading. So very small. And it just has to be a powder. It can be an API, it could be a blend, it could be an excipient, it could be an amorphous dispersion. What happens after you load the material into the cell is that solvent passes through the material, and a few things could happen. The compound could either dissolve. You could see, say, if it's a salt, if it disproportionates. We've seen particles go from a solid state into a liquid state and then crystallize back out. We've also seen a solid to solid transition. So all of these images are captured under a microscope with high content imaging. And what you're seeing is basically the particles dissolve with the video that's repeating over. And based off the rate at which it dissolves, it's all based on the Noyes-Whitney equation. You can then calculate what the dissolution rate is and what the solubility of that is. And there's a number of applications. We talked about spray drying. So with 100 micrograms of material, you can figure out what solvent best will dissolve the API. We can do things like the PC-SAFT or the Hansen solubility assessment to predict what excipients will work best for your material. We could basically run the simulated biological fluids to get the biopharm assessment, and we can even help with crystallization process development. Let's say if you're working, like you don't have much material, and your compound is highly soluble in some of the, basically your solvents that will dissolve before you cool down or add a co-solvent. You just need 100 micrograms to get that solubility information right up front. Next is the kinetisol process that was developed by Austin PX. It's an alternative way to make amorphous solid dispersions. Your drug substance is usually crystalline, it's insoluble, and poorly bioavailable. And then you have your excipients. It's all powder blended together using high shear granulation in order to render the material amorphous. You don't have to worry about dissolving your API in a solvent or the excipients. The throughput is pretty good on the commercial scale. It's up to 40 kilograms per hour. You don't have to worry about removing solvents at the end of it. You do add heat to it just based on the friction, but you're usually less than 200 degrees rises, and your material is only in contact for 10 to 20 seconds, so it's quick. Your output is an amorphous solid dispersion that behaves a lot differently than spray-dried particles. Spray-dried material tends to be fluffy, low density, tends to be difficult to handle. The kinetisol particles are dense. They could be directly compressed into a tablet. You don't need to do additional handling. And then you get the solubility increase as a bonus because the material is amorphous. I'm going to go over a few case studies. I think we're probably short on time, so I doubt I'm going to get through everything. But my colleague Steve Barrick and I, we have a booth in the room next door. I have a copy of the deck. Feel free to come on over. We could further chat. At the end of the talk, too, there's a QR code where you can scan and get a copy of the deck in advance. I'm at least going to cover the first two case studies, which highlights a collaboration between Austin PX and the Solubility Company that compares kinetisol particles to spray-dried particles, and then a collaboration with Microsize and the Solubility Company about basically watching a salt that disproportionates and trying to hit a particle size to capture its high solubility to get it to dissolve before it triggers a disproportionation event. The two areas I'm probably not going to get to are the continuous manufacturing from grams to metric ton scales using cojet milling as a way to not only micronize but blend materials, and then using a turbo screen to rehydrate material at the end. So the tool compound was from Boehringer Ingelheim. This is all published information in a joint collaboration with Austin and Boehringer Ingelheim. Essentially, they had a poorly soluble API, and they made spray-dried particles, and then they used the kinetisol technology. What happened was with the spray-dried material, it was dose dumping, and you can see on the left-hand side that the top curve, material is dissolving in simulated gastric fluid. Meanwhile, the kinetisol particles maintained intact, they didn't dose dump, and they were able to release when, during the two-stage dissolution experiment, switch over to FASF. And this bore out in canine studies where you had higher exposure due to the sustained release of the kinetisol particles compared to the spray-dried particles. And I'm going to show a few videos of what we did with the Solubility Company to show, basically corroborate the results and actually see what's happening. We ran the system at pH 2 and at pH 6.8. On the top row, we have the neat API and the spray-dried material. Under the pH 2 system, you could see the API slowly dissolving, so it has some solubility, but at pH 6.8, it's not dissolving at all. If you look carefully at the pH 2 buffer with the spray-dried material on the top corner at pH 2, you could faintly see like a little burst where there's a little bit of a dose dump. But what's really noticeable at pH 6.8, you just see a flash of material come through the video. It's quick. It takes like a second, and it dissolves. So there's your dose dumping as it transitions from pH 2 to pH 8. On the bottom rows are the kinetisol particles, where you can see they slightly swell at pH 2, but they behave totally different at pH 6.8. It's like it's an explosion of particles where the API is like slowly leaching out in a sustained release mode. So with this technology, you can actually visualize what's happening with your formulation and see visual evidence of what's going on. The second part is the race against salt disproportionation of an RDCS class 2 compound. So there is a poorly soluble, poorly bioavailable freebase. The customer tried to make salt out of it, which they were successful. It was 6 times more soluble. However, the bioavailability was not there. It was ineffective just because the material was disproportionating. So what we did was employ the SPA methodology, and we watched the particles dissolve, and we saw a 2-phase dissolution where we're basically able to capture the time where the HCl salt dissolved, but then we saw the lag phase where the material disproportionated. We're able to back calculate on what the particle size needed to be to make sure the material dissolved within time before the disproportionation event occurred. So we had a target on what particle size. We used a spiral jet mill to achieve that particle size. So anything less than 6 was able to dissolve fully. And then you can see with the curves, as you go to higher particle size, your basically your solubility tails off because of the disproportionation events. I'm just going to go over this really quick, just because the images are cool. This is spiral jet milling and how it can improve content uniformity by particle size reduction, as well as blending. So on the top left is unmilled material, unmilled API. We distinguish the two by using SEM and an EDS detector. So the green material is just silicon dioxide, and then the API is purple, it's HCl salt. So those are 2 elemental signatures, silicon and chloride. A lot of times people just focus on the API and micronize it and then tumble to blend. But you can still see on the bottom left that you see API rich and poor domains where the blending isn't uniform. Oftentimes people will jet mill the material separately, then blend. This could have flow challenges. And the blends you can see on the top right, which is excipient and the API. So you see black areas on the green and the purple images where basically you're not fully mixed yet. If you co-jet mill the materials together, the blending is superior. There's less regions that you see below with the co-jet milled. So it gives a superior blend over just separately jet milling and then mixing. We've done this on commercial scales where we employ continuous manufacturing, where we pre-blended the material in a ribbon blender and fed directly into a spiral jet mill. We did this on a compound that was being formulated for a DPI, where we had 2 active ingredients and 1 excipient. And we were able to basically scale this from 100 kilograms up to 250 metric tons. So we're able to move a large volume of material without having to put a major CapEx expense to run this in batch mode. And we got excellent data. And then finally, this is our controlled dehydration through humid air swept classification, which we use a turbo screen, where we converted a monohydrate to a dehydrate during the micronization. It dried out, and we were able to essentially rehydrate that material by humidifying the air in the room and then running it through a turbo screen, so each individual particle is exposed to moisture. And the existing solution before this was to just hang material in a super sack for a week, but it wasn't reproducible because moisture wasn't migrating into the super stack. With turbo screening, each particle gets exposed. So with that, I just want to say thank you for your attention and hearing me explain about the ecosystem that we're making at Microsize. And I'd like to thank all my colleagues at Microsize, the people that I work with at the Solubility Company, Austin PX, as well as Solumatrix. And thank you for your attention. And this is the QR code that I mentioned.
Thank you, Jason. We're going to take a quick break, but any quick questions for Jason before we... Yes.
Hi, really nice presentation. I'm Brian from Avonic. I was curious what that solubility company technology you showed. Is it possible to put in polarized light or something like that to kind of visualize what would be happening in real time with that technology? It's going to be cool. You know, I saw this previous presentation on the whole liquid-liquid phase separation thing. If you had ASDs, it would be really cool to see that in real time with a very small quantity of material, any potential recrystallization or something like that.
Yeah, it's something that's not currently available, but it's things that we're talking about.
Okay.
We're also wondering, can we actually incorporate like a Raman spectrometer to get morphology, polymorph data too. So not with this current generation. Unfortunately, that's all offline, where we could do a small slurry experiment and look at crystallization either under a PLM or XRD.
Very interesting. Thank you.
Thank you. Any additional questions, Jason? No? Okay, thank you again, Jason.
Thank you.
Obviously, Steve's in the corner as well if you want to